Divya. Today's topic is replication in eukaryotes. So first, definition. So the process of making an identical copies of a duplex DNA, double-stranded DNA, using existing DNA as a template for the synthesis of new DNA strands. DNA replication at heterochromatin region and telomeric region is slow, while at euchromatin region and centromeric DNA replication is faster. So chromosome in eukaryotic cells consist of DNA, protein, some chromosomes, RNA. This is a cell cycle. So in G1 phase is preparing for DNA replication, and in S phase DNA replications occur, and G2 phase a short gap between mitosis, and last phase M phase. So mitosis and cell division in this occurs in this phase, M phase. This is a cell cycle. So this is a center dogma model. It is proposed by Crick. So DNA transcripts RNA, RNA translates into protein. So this is the center dogma model. So semi-conservative applications, conservative applications, dispersed replication. So that is the original DNA, and after one round of replication, this semi-conservative replication. So experiment of DNA semi-conservative replication. First, we discuss about the differences. What is the difference between prokaryotic replication and eukaryotic replication? So first, locations. So in prokaryotic replication occurs inside the cytoplasm, and in eukaryotes occurs inside the nucleus, and in the origin of replication. So one origin of replication per molecules of DNA, and here in eukaryotic replication have many origin of replication in each chromosomes. In ori length 100 to 100 more nucleotide in length, and in eukaryotic replication about 150 nucleotides is the ori region, ori length, and initiation elements carried out by protein DNA A and DNA B in prokaryotes, and carried out by the origin recognition complex in eukaryotes, and the DNA polymerase first, second, third is found in prokaryote replication, and in eukaryotic replication alpha, beta, gamma, delta, and epsilon is that type of a DNA polymerase found in eukaryotic replication. Uh, Difference between prokaryotic and eukaryotic DNA replications. So here we are showing in this image that thing. So basic rules of replications. First thing, semi-conservative starts at the origin, semi-discontinuous synthesis always in the 5' to 3' direction. RNA primer required in humans occurs at the rate of 3000 nucleotide minutes. In bacterial cells, it occurs at the rate of 30,000 nucleotide per minute. So DNA replication machinery involved in eukaryotes: DNA polymerase, alpha, delta, epsilon, gamma, helicase, topoisomerase, primase, ligase, SSB, single strand binding protein, DNTP, dinucleotide, triphosphates, magnesium, and ATPs. So what is the role of protein? DNA helicase. So unwinds the double helix, RNA primase, synthesize RNA primer, single strand binding protein, keeps the two strand separated. DNA polymerase alpha add RNA primer during initiation. DNA polymerase delta proofreading of DNA and add Okazaki fragments. DNA polymerase epsilon synthesize DNA, and another work is done by proofreading. So, and the last protein is in replication DNA ligase that joins the end of DNA segments and does DNA repair. So first we discuss DNA polymerase alpha involved in initiation. First thing, synthesize an RNA primer, then adds DNTP, dinucleotide triphosphates, and after that a complex of 4 subunit 50 kd and 60 kd are primase subunits, and 180 kd Subunits DNA polymerase synthesize 10 to 12 nucleotide RNA primers. So that synthesize RNA primer for the leading strand and each lagging strand fragments. So DNA polymerase delta. So the principal DNA polymerase and add Okazaki fragment in lagging strands has role in proofreading. This is a consist of a 125 kD and 50 kD subunits. So this 50 kD subunits interacts with the PCNA. PCNA proliferating cell nuclear antigen. So next is DNA polymerase epsilon. So this does catalyze replication of leading strand, also contribute in proofreading. So DNA polymerase epsilon consists of more than one subunit. Polymerase delta and epsilon both contain the 3' to 5' exonuclease activity required for proofreading. And DNA polymerase beta. So this is DNA polymerase beta monomeric having 30 to 38 kD involved in DNA repair process. And last one is DNA polymerase gamma. So then DNA replicating enzyme of mitochondria, DNA polymerase gamma. So protein involved in eukaryotic DNA synthesis, PCNA, proliferating cell nuclear antigen, provides substrate for DNA polymerase delta, the eukaryotic counterpart part of the sliding clamp of E. coli, PCNA, also encircles the double helix. Next, RPA, replication protein A, single-stranded DNA binding protein that facilitates the unwinding of the helix to create two replication forks, the eukaryotic counterpart of the SSB protein. And the next, RFC, replication factor C, the eukaryotic counterpart of the complex clamp loader of E. coli that is loads PCNA on DNA. Next, ORC, origin recognition complex, multi-subunits protein binds to sequence with the replicator interacts with the two other protein CDC6 and CDT first, resulting in loading of MCM, mini chromosome maintenance complex, on DNA. So that MCM, mini chromosomes maintenance complex, that are heteroisomer, MCM2 and MCM7 ring-shaped replicative helices. Once the mini chromosome maintenance proteins have been loaded onto the chromatin, that are ORC and CD6 can be removed from there. So DNA ligase, they are classes of enzyme that catalyze the formation of alpha phosphodiester bond between two DNA chains. So that enzyme binds hydroxyl group at the 3' end of DNA strand to phosphate group at 5' end of the others. So linking the two different fragments and completing replication of the region of the lagging strand. So the DNA replicase consists of three stages like initiation, elongation, and termination. So during initiation, proteins binds to the origin of replication while helicase unwinds the DNA helicase and two replication forks are formed at the origin of replication. During elongation, a primer sequence is added with complementary RNA nucleotide, which are then replaced by DNA nucleotides. During elongation, the leading strand is made continuously while the lagging strand is made in pieces called Okazaki fragments. So during in the last steps of replication, like termination, primers are removed and replaced with new DNA nucleotide and the backbone is sealed by DNA ligase. So first we discuss about initiation. So to accomplish this initiation is parted into two temporally discrete steps like pre-replication complex formation during G1 phase, activation of pre-replication complex by CDK, and